Hello student, good afternoon. Today I will start the new topic cell division. Cell division. There are three methods of cell division, namely there are as follows: amitosis, mitosis and meiosis. That is the three types of cell division: amitosis, mitosis and meiosis. Amitosis means what? What is meant by amitosis? This is the simplest division in which nucleus elongated becomes dumbbell shaped and finally divides into two along with cytoplasm. Thus, the division of chromatin is not precise or definite. It occurs in WBC, T generating cells, and in some protozoa. That is called as amitosis. Second is mitosis. In mitosis, four phases: prophase, metaphase, anaphase and telophase. In mitosis, cell division takes place in the body cells or somatic cells. It occurs both the somatic and germ cells. It is a complex process of cell division which involves an equal divisions or nuclear chromation to the two daughter cells. It has been observed in almost all cells except those of testes and ovary. It may be defined of the process whereby the chromosomes become duplicated longitudinally into equivalent parts which separate to opposite poles of the cell. The main divisions of this cycle are prophase, metaphase, anaphase and telophase. Cytoplasmic separation of two daughter cells is known as cytokinesis. First phase is prophase. Pro means before. In the beginning of prophase, the chromosome appears as thin thread-like then they starts condensing by a process of coiling and folding. Each chromosome is composed of two chromatids which become shorter and thicker, and the primary constriction containing centromere becomes clearly visible. In the rarely prophase, chromosomes are evenly distributed throughout the nucleus, but in late prophase they move towards the nuclear periphery. The nucleolus disorganizes and is absorbable in the nuclear organizer at the end of prophase. In the cytoplasm, spindle is formed between the esters and centrioles that move towards the poles. Centrioles replicate at interphase during the spheroids. Centrosome, nucleus, chromosomes duplicate in G1. Spindle fibers, early prophase, middle prophase and late prophase. Metaphase. Second is metaphase. In the beginning of metaphase, prometaphase, the nuclear disintegrates and there is mixing of the nucleoplasm with the cytoplasm. Chromosomes get attached to the spindle fibers and are oriented at the equator plate. The spindle nuclear envelope, spindle metaphase, prometaphase. spindle fibers are in the equatorial. Spindle has continuous microtubules and fibers arising from the chromosomes are known as chromosomal fibers. The spindle apparatus which help in the arrangement of chromosomes at the plate. Anaphase. Third phase is anaphase. After the spindle is formed and the chromosomes have arranged on the equatorial plate, the chromosomes split at the position of centromere. Also, sister centromeres separate from each other. In anaphase, the chromosomes attain characteristic shapes depending upon the position of centromere. The chromosomes by V-shaped, J-shaped and I-shaped in metacentric, submetacentric and acrocentric chromosomes respectively. The spindle fibers shorten 1/3 to 1/5 of the original length. Last phase is telophase. In telophase, chromosomes again uncoil. After the anaphase, chromosomes have reached the poles. The nuclear membranes is reconstructed around each group of the chromosomes, giving rise to one nucleus to each pole. The nucleoli also reappear at the stage. Cytokinesis is the process of separation of the cytoplasm in animal cells. There is a construction at equator that finally results in the separation of the daughter cells. The division of one nucleus into two is often called karyokinesis and is followed by cytokinesis. The cytokinesis is the division of the cytoplasm and into two cells and can be brought about into two ways. Namely, these are as follows: cell furrow, cell plate. In case of animals, the outer layer are more flexible due to the absence of cell wall. In such cases, a circular constriction appears at the equator and it converges on all sides, finally separating the two daughter cells in the following diagrams. Cytokinesis by cell furrow. In plant cell, a more rigid cell wall is present. In such cases, a cell plate is laid down. This cell plate generally initiated at the center and is completed towards the peripherally in the below diagram. Nucleus, cell plate and cytoplasm. Cytokinesis through the formation of cell division, initiation of cell plate. Cytokinesis, one cell divided into two daughter cells in the telophase. Okay, thank you.